Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members and the broader scientific community about topics related to antibody discovery and development. Our expert speaker, Dr. Gang Lu, will be discussing AbDrop Speeds, accelerating your antibody discovery with single B solution with us today. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn the show over to our speaker.
Hello, everybody, and thank you, host, and thank you for what you and the Antibody Society has done to make this webinar happen. So again, my name is Gang Liu from Biointron, a CRO company that offers antibody discovery and antibody production service. So let me play my slides first. I hope everybody can see my slides and my voice can be heard, right? So as this webinar title shows, I'm going to introduce a single B antibody discovery solution called AbDrop that's built by Biointron, and I will show you why this antibody discovery solution is unique and how it can help you in your antibody discovery project. So my webinar will be composed of three parts. So first we'll talk about something about the major antibody discovery approaches. Inside this, we will talk about the advantage of a single B-cell antibody in antibody-based research, why it's unique. And secondly, I will introduce the Biointron single B antibody discovery solution called AbDrop. To put it simple, it's a microfluidic, technology-driven, droplet-based single B-cell screening antibody discovery platform. So my webinar will be ended with some case study. In this case study, we will see that this AbDrop antibody discovery solution is very powerful to upgrade and accelerate your antibody discovery. First, let's take a quick look about the general antibody discovery workflow. Actually, no matter it's hybridoma, as shown here, or phage display, or single B-cell screening we're going to focus on, they kind of follow the very similar workflow. First, begin with animal immunization, and then you harvest B cells, followed by antibody coding sequences obtaining. When you get this antibody coding sequence, you can do anything like antibody production and antibody function screening and more. So the most tricky part will be the one highlighted in green, because as everybody knows, I would not say it's impossible, but it's very hard to culture primary B cells in vitro for very long, maybe days or 1 or 2 weeks, but that's not enough for us to get the antibody coding sequences. This is exactly the case for solution like the hybridoma. This is why we need the solution like hybridoma or phage display or single B-cell screening. For hybridoma, what people do is that they hybridize, they fuse the primary spleen cells with the myeloma cells to make these cells immortal. So by doing so, they keep these cells. Then they have a huge, wide window for them to get the antibody coding sequences. But while people making this hybridoma, it's certainly true that we will have to lose, I would say, plenty of, in many cases, most of the B cells. Say only 10% B cells survived, so you can only get the best antibody hits from the only 10% left B cells. So this is, and also hybridoma solution is time taking, is the labor is time taking, and it's labor cause much of labor work. For the secondary, the second one, phage display, is kind of a similar, is time taking, and what people do to keep this antibody coding sequence is that they display these sequences on the on phage. By doing so, they keep these antibody sequences. So it will allow them plenty of time to do panning, screening, finally get the sequence they want. Finally, single B. So with single B cell sorting, I would say in days, we screen each B cells individually. In like one or a few days, people can finish screening all of those primary B cells. And after that, even though we lose all of the B cells, that's fine because we already got these antibody coding sequences. Okay, this is a general of the so far majorly used antibody discovery approach. So if we dive a little bit into the single cell segregation-based screening technology, I would say, although in the past, in the recent years, plenty of approach, single B cell screening approach has appeared. They actually share similar workflow. Like first is B cell compartmentalization, and which means you have to put each one single B cells in a very limited room, then followed by B cell culture for maybe a few hours for the antibody to be produced and released into this small room, and finally by scanning and read the signal and sorting the target B cells. And finally, you will get those target B cells, and followed by something like NGS to get the antibody coding sequences. For each single cell screening technology, I would say the most critical part is in the step one, is compartmentalization. So you can either put the single cell into one microchamber, or you can generate a microdroplet in where the single cells is inside, along with some other assay reagent, or in a so-called lab on the particle. So our AbDrop antibody discovery solution actually is based on the second one. It's microdroplet based. So I will touch to this part after this slide. So why we say single B screening in antibody discovery is advantageous. So we can tell from these points. First is speed. So as I said, with hybridoma making and screening generally take like 3 months or longer. For phage, maybe not that bad, but still library construction and panning, screening still just take like 2 months or even longer. But with single B cell screening, just like days, like 1 to 3 days or 1 week for B cell sorting and NGS and screening. This is the speed. So that's what we call single B cell screening is a faster track. Secondly is the antibody diversity. I would say, for me at least, I care about antibody diversity very much at the very early stage. With hybridoma, as I said, you may lose plenty of B cell clones, or in many cases, most of them. So you have to find the best one from the remaining B cell clones. That's why its antibody diversity is very limited. This is for routine antibody discovery case should be fine, but it will not for some antibody discovery project where advanced function is required. And for phage display is kind of better, but still very limited by like library constructing, by its library size, and more. This is antibody diversity. For antibody maturation, I would say both hybridoma and single B cell screening, they are both fine because they both are screening the primary mature B cells. But for phage display, kind of less because you have to display the antibody sequence on phage. So from a phage, which is a non-mammal system, to mammal system, there may be a gap. So after you get antibody sequence, you might have to do some further optimization to make this antibody more mature, more stable, and so on. Finally, is through output. I would say a single B cell screening is a very high through output. I will show you why. And with hybridoma, it's super low, and it's time taken. And for phage also is low, although it's not as low as the second one, I mean hybridoma. So now we enter our second part. I'm going to introduce Biointron's AbDrop to see how AbDrop works. To put it simple, it's a microfluidic, technology-driven, droplet-based single B cell screening platform. So as a first step, we need to sort to get the target B cells. So after animal immunization is done, we will harvest the B cells, and then we load both these B cells and some special bioassay reagent to generate microfluidic droplet by the so-called cell encapsulation. So as shown in the green, in this image, the bigger green events, they are microfluidic droplet. Inside it, they are cells. I mean, one single B cells and some bioassay reagent. And after this microfluidic droplet is formed, it will be transferred to be cultured for a few hours. Although the antibody it may produce in very low amount, as I said, since the room is very limited, so the antibody amount is still high enough for us to detect. Then we will do the droplet scanning and sorting by assay called FRET. So I may not go very deep in this so-called FRET. To put it simple, which is that when the target antibody is produced, it can be recognized by its target antigen, the green events, followed by the secondary antibody, which will bind to the Fc part of this target antibody. It's only when this tri-complex formed will the FRET happen. So when this FRET happen, as the red flash show, we can detect the fluorescent signal, so which is shown like in red. So when in case there's no cells in the microfluidic, so the events will turn like empty, like gray or dark. But if there is antibody that's produced but is not target one, so we can only detect the donor's signal. Donor signal only with green. So either green or gray ones, they are all bad ones. They will be treated as a waste, and so only the red ones will be sorted for the further analysis. So after before I go to the next step, I will pick up 3 details about how this labeling sorting is done. So this slide shows the how droplet generation is done. So as I said, we load the both cells and bioassay reagent. So the cell concentration we set it as 1 million per mil. So only at this cell concentration can the one cell in one droplet will be formed. If with higher, we found that they are not good. So at 1 million cells per mil, single cell encapsulation will happen. So after this microfluidic is transferred to so-called cultivation chamber and cultured for a few hours, we will stain and detect this secreted antibody with either directed antigen labeling. This antigen is shown as the green dot. As you can see, so the antibody is recognized by its target antigen shows as gray dot, and then the antibody, a secondary antibody that in blue will recognize Fc part. So by forming, after this tri-complex is formed, the threat will happen. Then so we will scan the signal and detect the threat signal. In case that there's no free antigen can be used for, say we have some antigen target. They are not protein. They are multi-transmembrane proteins. We have to use cells. So in this case, this antigen target is located in the cell membrane. So working in a similar way, so the membrane bound antigen will be recognized by the secreted target antibody, followed by the secondary antibodies bound to this FC part. So after this complex formed, so the threat signal will be detected. So then we will sort all of those positive B cell hits as a whole. See the red one, the green one and empty one, they are all treated as West. Only the red dots, red zone will be sorted as a pool. Then we come to the step 2. In step 2, after we got all of those target B cells, which may be possibly producing the antibody that we want, we will pair each single B cells with one single barcoded bead. This bead is kind of unique because each bead carries a pair of so-called barcode. Actually, there are just some DNA sequences followed by fused to like poly A. So this barcode works as primer, a pair of primer to be used in the following steps. When this one cell paired with barcode beads is done, the B cells will be lysed, and then this mixture will be retrieved by a magnetic bead field, followed by the RT. Then we will get the cDNA. So after we get all of cDNA from each B cells, we will do the NGS, followed by some bioinformatics analysis. You may wonder how we can tell if the heavy chain, light chain, they are from one single B cells. So as I said, each single bead, their barcodes, a pair of barcodes, they are of the same sequence. So if the heavy chain, light chain, say here, heavy chain cDNA, light chain cDNA, they are of the same barcode sequence, we would say, okay, this heavy chain and light chain, they are from one single B cells, so they are naturally paired. If not, we will just dismiss it. When we get those paired sequence for each antibody hits, we will begin with gene synthesis and polymerase preparation and two or hex cells transfection, and then antibody production, purification. This will be done just in 1 week. After we get this purified antibody hits, we can do many of the downstream things like plate-based screening, cell-based screening, affinity analysis, and many more functional screening, which I will show in my coming slides. Okay, so in the coming 10 to 15 minutes, I will share you by 9 case studies. From these case studies, you will see Biointron's AbDrop is this single bead antibody discovery solution holds very, very unique key features that can separate it from any other traditional antibody discovery solution. First is a fast track approach and of high efficiency. Why is so fast? Because it can screen 1 to 2 million cells per time per loading in 1 day. So say we finish the animal immunization. We harvest B cells from spleen. We prepare the cell suspension, and then we load up to 2 million cells into one chip. And then in just one day, we can finish the scanning, sorting of all of those 2 million cells just in one day. Okay. After, in just 1 week, we will get the paired sequence, because the NGS and the bioinformatic analysis maybe take a few more days. So, but just in 1 week, we will get paired sequences of each antibody. And from sequences to purified antibody, as I just showed, is just in 1 week. So in general, from shots, I mean from the first round of animal immunization, from first round of animal immunization to verified hits with the Biointron's AbDrop, just take like 2 to 3 months. So compared to hybridoma, is a very fast track, because for hybridoma generally it takes like 6 to 7 months or longer. For phage, also may like 5 months or longer. So this is the first fast track approach of high efficiency. Secondly is actually this point I like the most because it can maximize the CDR diversity by mining. I may not say entire, but the most plasmid cells we can get by scanning each individual B cells from the whole entire plasma cell pool. We can try to keep all of those B cells. So thus we will get the maximal diversity of those CDR sequences. With these CDR sequences of good diversity, we can have a lot of antibody to choose from, to compare, to get like positive controls, negative controls, do many functional screening, affinities, affinity analysis, and more. But say with hybridoma, you just got limited amount of hits. You will not got a chance to do so many things. And also, this is a very good. It's a very good solution for some challenging targets, with which the immune response is very weak, thus leading to very limited B cell clones. Or in some antibody campaign, you need some advanced antibody function like super high affinity or unique binding site, or like blocking or some other functional requirements. So in this case, you have to look at a much bigger pool to find the dream hits. Okay, the second. Third is, I would say, for our Biointron AbDrop antibody discovery solution, is good not just for mouse as a host. It's also good for rat, rabbit, and transgenic mouse. We're now working on more species like human PBLC and also some other bigger animals. Finally, so as other single B cell solution can offer, this Biointron AbDrop antibody solution, it mines primary B cells so that you can get natively paired sequences. Okay, in the coming slides, I will show like 9 case study, so which will offer some fact to support these key features. Okay. So first one, the target I would say is kind of a routine. It's a single pass transmembrane protein. We use a mouse to raise this antibody. So after B cell sorting and NGS, we finally got 446 paired sequences, and we choose 62 of them to express. And finally, we got 59 ELISA binders. So I would say you can tell that compared with the benchmark, all of those hits, I would say most of the hits we got, they are of much better binding activities. And also, if you look at the affinity, similar story. So compared to benchmark, its affinity is most of them reached -10. So in addition to binding, I would say in many cases people more care about the CDR sequences diversity, as I just talked about. You can see that their CDR diversity is very good by looking at its phylogenetic tree. It's very diverse. And also its binding epitope also is very diverse. As shown, the orange zone, they are antagonists. And the green and blue, they are agonists. While the yellow one, there are some antibody hits that fall in between. In many cases, even though say you just need the hits from the orange zone, say you only need an antagonist. But in your early stage, that you might have to use some agonists or some neutral ones to compare to make sure the functional antibody are real the one you want. This is the first case. Second case is kind of unique because the target now comes to a GPCR. So as everybody may or may not know, GPCR is kind of unique compared to the single pass transmembrane protein because this kind of protein are hard to get of this material as proteins. Secondly is that GPCR and proteins mostly share their sequences between mammals like human, cyno, rabbit, mouse, rat. So this will lead to very low immune response. As I said, so you may get limited B-cell clones. Even though you keep all of those B-cell clones, you have just little chance to get your dream hits. So in this case, since as I said, there's no protein antigen that's available, we tend to DNA and the cells to immunize mouse. But still, finally, we get plenty of cell binders. So as shown in the fact screen. From the 177 pair sequences, we choose 78 to express. And among the 78, we got around 30 cell binders. And we're so lucky is that among these 30 cell binders, 6 of them are blockers. So 6 of them, they are functional ones at cell levels. And also, as you can see, there's no benchmark for this target because I would say people tried a lot with high hybridoma but failed. So with mouse. So in this case, there's no benchmark as positive control for us to. But finally, we got a lot of his FACS binders and blockers. Cas3 actually is also a GPCR target to raise antibody from mouse. This time we changed our antigens from DNA and cells, which I would say actually don't work very well. We replace this DNA and cells with M and LNP. This form of antigen work much better. So for this target, finally, we get 254 paired sequence, and we choose 70 of them to express. And finally, from this 70 case, we got in cell-based level, human cell binders 25, and among these 25, we got 20 that are cyno target binders, And what's amazing is that we got 13 mouse target binders. This is amazing. Why I'm saying this amazing, because this antibody is raised from mouse, so it's I would say it's pretty low chance you will get an antibody from mouse as a host that can also cross react with mouse target. But we did, and we got 13. So you can imagine. So if with hybridoma as shown in case 2, there I will not say 0, but there will be very little chance for we to get like any hits. Actually, I have my own story to share here. I once worked with some GPs targets. It took me, believe it or not, 5 years. But finally, I only get like a few hits. So, and I use the hybridoma. So that's why hybridoma, as we said, is low efficiency, time taking, and to work very bad for some challenging targets. The next case is kind of unique because of its size. It's just a peptide. It's a 44 amino acid long peptide. So as we all know, for smaller antigens, either protein or non-proteins, because they are not large, because their special structure is not that complex, so their immunogenicity is weak, is low. So in this case, if you use hybridoma, as I said, so with such a low immune response, and plus you may lose plenty of the B cell clones, there may be weak chance for you to get very good hits or any hits. But with our AbDrop solution, you can see we get plenty of cell binders, and many of them are of biological function. So as shown in this table, 417 paired sequence were obtained, and we choose 51 to express, and 37 of them, this may be a mistake, it's not ELISA. Yeah, it is. Yes, it is. It's right. It's 37 of them are positive in ELISA test, and among the 37, 17 of them by cell-based screening, they're blockers. The next study is to raise antivirus protein, but this time around not from mouse, but from a rabbit. Because in some cases, people turn from mouse to like rabbit or rat for some special goals, like they want this antibody to be of much higher affinity, or to recognize some special size, like a PTM size, which the mouse antibody may not work well. So in this case, we use the rabbit as the animal host to raise antibody. The benchmark we used is a mouse antibody from hybridoma. So as you can see, with the rabbit and our AbDrop, totally we got 391 paired sequences, and we choose the 22 to express. And among these 22, 19 are good ELISA binders. I would to mention that compared to the benchmark, which is a mouse antibody from hybridoma, all of our hits, they are of much higher binding affinities. If you can, as you can tell from these numbers, our hits are like 5 to 10 as strong as the positive control in binding the target. The next case is also to raise some antibody from rabbit. But this case is, I would say, is different, because this time the antigen is not protein anymore. So as we know, with protein as antigens, even though the protein is of a smaller size, but still their immunogenicity is still kind of strong enough to trigger immune response for our downstream B cell screening. But with small compound, the immune response is super weak. So actually, in this case, we conjugate this small compound. Actually, it's a payload from ADC to some carriers like KLH or BSA. And then after animal immunization and our AbDrop B cell screening, we got, as shown here, 1,372 paired sequences. Okay, here you may wonder, in general, how many B cell hits and paired hits we may got. So here I can show some general information. After B cell sorting, we can get generally thousands or maybe tens of thousands B cell hits. So after NGS, we may get thousands or hundreds paired sequences. And then we will discuss with our customer to see which paired sequence they may choose to be expressed. So based on their CDR sequences, also like some PTM liability hotspots or anything else, they will choose working along with us, like tens of them or more, to be expressed for the downstream screening. Okay, back to this case study. So even though it's a small compound, with our AbDrop antibody discovery solution, we still get more than 1,000 paired antibody sequences, and we express tens of them. Among these tens of them, as you can see, 29 of them, they are of a very good target bindings. Okay, then go to next case study. Is not that unique, but I just want to mention that this time's not mouse, not rabbit, but rat. So just to show you guys that the AbDrop single B antibody screening platform is good for mouse, rabbit, and rat. And also, as you can see, the affinity is kind of high. They reached 10^-10. So the last two case study, the animal host is kind of different, because we use fully human. Actually, it's a transgenic mouse to raise antibodies. Because for many of our customers, if they want to develop some therapeutic antibodies, in many cases, we have to humanize them after we get some good hits. But by doing humanization, some risk will be introduced that may appear in stages like CMC or preclinical or clinical stages. So better we got some so-called fully human antibody from transgenic mouse. So this transgenic mouse model called HugoMap is from our partner called Cyogen. They developed this transgenic mouse by replacing mouse heavy chain large chain regions with human ones. Okay, now back to this case study. So the target is VEGF. So with our AbDrop antibody discovery solution, we after animal immunization, we got 292 paired sequences, and we choose some of them to express. So you can see 13 of 20s that are expressed. They showed very good binding activities. Last but not least is the case study. This case we still use the transgenic mouse. In this case, the antigen we call it antigen X, which is actually the human protein. This protein shares a high sequence similarity with mouse. It reached like 90%. To be honest, in many of our finished project, some human proteins target they share even higher, like 95, 96. So, but still, so back to this case study. So this target was tried by someone else elsewhere with hybridoma, but in that case, only 2 binders were obtained. Okay, so but our customer want they ask for more because with this antigen and to immunize animals to get antibody, they want to develop therapeutic antibodies, so which means they need plenty of more hits to compare, to study, to choose from. So then they turn to us. So with our AbDrop and single B antibody discovery solution, we were able to obtain, as shown in the slide, plenty of specific binders. So we choose 34 antibody, and as shows in the curves, 28 of them showed very strong targeting binding activities. Okay, I think this should be the last case study. So I'm going to end my seminar with this summary. So as we have seen, so single B cell technology is compared to traditional ones, the up-to-date antibody discovery strategy that can overcome limitation encountered by traditional antibody discovery method, like time-consuming, labor-intensive, very poor the CDR diversity, and not able to meet advanced functional requirements. A lot of trouble in some non-protein targets, and many more, and also very bad for some very challenging targets. But with single B solution like Biointron's AbDrop, we can do that. Second is that, so by introducing our AbDrop in Biointron, we can see it's a microfluidic, technology-driven, droplet-based single B-cell screening platform. So as shows in the case slides, and many more, maybe you can find out from our website. So it's a very powerful tool to not just accelerate, but also upgrade your antibody discovery research. Why I'm saying it also upgrade, because sometimes I would say time is not the first thing we worry about. In many cases, we worry more about antibodies quality. Okay, so back to these points. So with our AbDrop solution, every year in the recent 2 years, we can finish around 100 projects. And this number is still growing. And also our Biointron AbDrop antibody discovery solution, as shown, is good for multiple hosts, like mouse, rat, rabbit, and transgenic mouse, and more. And also the target we have covered are like proteins. These proteins we have been very successful in segregated protein, like cytokines, and single transmembrane proteins, multiple transmembrane proteins like CD20, and 7-time transmembrane protein like GPCRs, and many more. And we also the target also covered a small peptide and non-proteins like small compound toxins. And as for the antigens, we have tried proteins, DNAs, VLPs, cells, and most recently mRNA LNPs. And now I would say we are experienced in very challenging targets. Okay. Finally, I'm going to end my talk by mentioning some information about Biointron. As I said, Biointron is a listed CRO. We have more than 1,000 overseas customers. We offer antibody discovery and antibody production services, as shown here. For antibody discovery from gene sequences to purified antibodies, generally just take 2 weeks. And also, we are very good at bispecific antibody production. So far, we have very good experience in more than 400 different formats of bispecifics. Every year, we can deliver more than 1,000 bispecific antibody production project. Also, we offer fucosylated antibodies production for you to get some advanced ADCC effect of your antibody drug. And if you have plenty of antibodies to compare to express, but for each of them, you don't need much big amount. We have a service package called a rush map. So we can finish rush map. We can handle like 5,000 samples for each single batch in like 2 to 3 weeks. So yeah, and also some non-antibody protein production. As to antibody discovery, we have just talked a lot about a single B cell screening with mouse, rabbit, and transgenic mouse from our partner Cygen. We are also very good at VHH antibody discoveries. We have our own farms. We have hundreds of animals, and for our customer to raise VHH antibody. Now we are also very good at antibody optimization and hybridoma sequencing. And if you have already narrowed down your antibody drug to like 1 or 2, we are good at two-cal stable cell line development. Okay, I think that's pretty much what I'm going to say. If you have any question about our Biointron service that we can offer, just call, scan, or visit us to find out how we can accelerate your antibody discovery. Thank you.
Thank you so much, Dr. Liu. And we have a few questions in the chat, so I will jump right in. So I'm going to start at the top. Why was VEGF the target for the transgenic mouse over the other targets, or over other targets in case study 8?
Why was VEGF the target for the treatment over other targets? Okay, so actually this is the decision made by customer. So they try to get some antibody that targets against VEGF for their downstream antibody drug project. So maybe, as I said, people don't want to do humanization of animals raised from non-human, because that may introduce some risk like ADA, or some CMC risk, or some physicochemical risks that may cause some surprise in antibody stability, antibody production yield level, or they may pose some challenges in antibody purification, antibody formulation. So people will try to avoid to do extra or to do too much in vitro engineering to antibodies that they read from animals. So with the transgenic mouse, we hope that the least in vitro engineering will be done. So thus we can reduce or avoid those risks that I just touched.
I see. Okay, thank you so much for that. And then the next one is, What was the process for developing the AbDrop solution?
Oh, that's a very good question. Actually, this AbDrop solution, we purchased the machine, the device from a company called Sphere Fluidics. They offer this type of machine is called a Satmine system. So, but merely with this device is not enough. So with some biocell reagents offered from them, some of them, but not all of them, and chips, we have to develop we have to figure out the know-hows to get our own reagents based on each targets and to build this whole workflow. So with their device, with their key reagent, with that key kit, so as I'm showing in the workflow, we have to immunize. We begin with animal immunization, and then we get the B cells. Then we load the load the B cells along with the bioassay reagent to finish the formation of the microfluidic, and then followed by microfluidic culturing, sorting, and then NGS. Get the sequences, antibody expression, and finally followed by the downstream screening. So actually, this was the process for developing the AbDrop solution.
Thank you for that. And so from the acquisition from the other company until the stage that you're at now, how long did that take?
How long did it take for us to establish the AbDrop solution?
Yeah.
I would say, believe it or not, it takes much long time. I mean, still not, right now, we're still working on it to facilitate, to further improve, to upgrade its capacity, to improve, to enhance its efficiency, to try to avoid some breakdowns. But exactly how long we spent from zero to for this solution to be ready for service, actually, I don't have the exact number for me because I'm kind of new for Biointron. I joined the Biointron in just 1 year, probably months, I would say. Yeah.
Gotcha. Okay. Thank you so much. Then the next question, you shared the benefits of single B cell screening over hybridoma and phasing. What are the scenarios where using single B cell screening is advantageous?
Good question. So say if you just to raise some antibody as true antibody or as some like flow reagent, you ask no about its function, its binding site, or you don't care about its diversity, you are good to go with hybridoma or phage. But in case, say, you have a very challenging target, with which the immune response triggered will be very low. Say you immunize animals with this low immunogenicity targets, nothing happens. Nothing happens. So even though you harvest B cells and don't forget with hybridoma, you will lose most of the B cell clones. It's possible you get nothing. So okay, this is case one. Case two is that even though you get plenty of antibody hits, what if you want some hits that can bind some hidden site or bind some unique site like the phosphorylation site? This cannot be done by antibody raised from hybridoma. I mean, may not be done. Or say you want some antibody of very high affinity. But with hybridoma, you say you only have 20 to choose from. You may already lose the better ones. So I would say, as I said in my post, big dream deserves big room. So if you want your dream antibody hits, you need to find it from a big pool. So, right? So the biggest fish in a pond is not a king. You must find the biggest fish from ocean.
Absolutely. Okay, and then I think the last question that I see is, why did you use the mouse as the benchmark when you used rabbit to produce the antibodies? Is there an advantage or disadvantage to that strategy? I think that's from case study 6.
Good question. Okay, so by saying benchmark, actually benchmark work both as a positive control, because when you do the plate-based or cell-based screening, you need to make sure your assay works. So that's why first we need a positive control to set up this test to make sure it's functional. We will not get false positive signals. Okay, this first. So in many cases, as long as this benchmark works good enough, it's good enough. For second, we call it benchmark in many cases because we want to beat it. This is exact case when you raise some antibody for drug. Okay, so that's what we call it benchmark. Okay, so it's kind of let's back to the question itself. Why we use mouse as benchmark? Because in that case, our customer want to raise antibody from rabbit for like advanced functional requirements, or for the antibody to recognizing some tricky hidden epitope. But rabbit antibody as benchmark or as positive control is not available. So the way we can get is from mouse. Yeah, actually the reason is simple.
That's
simple. As you can see, in some case study, there's no benchmark. So still we have to, because there is always so-called first in class. You will not always go with the project with a benchmark, but you still have to go first.
That makes sense. Mouse is a popular model, so it's readily available. So that's really
so when you turn to red, you will find no benchmark from rabbit.
Yeah. Okay. That makes a lot of sense. All right, great. That's the last question that we have. So thank you so much to our active audience for giving us some questions. And thank you again, Dr. Liu, for this insightful presentation. The on-demand version of this webinar will be available really soon, and a link will be sent by email to everyone who registered. Feel free to watch this or any of our other on-demand webinars when it's convenient for you on our YouTube channel at the Antibody Society. Thank you again, and have a great rest of your day.
Thank you. Thank you for everybody who come to attend this seminar. Thank you.